Human excretory system. In human beings, excretory system consists of a pair of kidneys, a pair of ureters, a urinary bladder, and urethra. The nitrogenous end product in man is urea. Kidney is a bean-shaped, red-colored organ. Kidneys are attached to the dorsal body wall on either side of vertebral column in the abdominal cavity. Each kidney of an adult human measures 10 to 12 centimeter in length, 5 to 7 centimeter in width, 2 to 3 centimeters in thickness, with an average weight of 120 to 170 grams. Its outer margin is convex and the inner margin is concave. The notch present on the inner margin is concave. The notch present on the inner margin is called hilus. Renal artery, which brings blood to the kidney from heart, enters the kidney through hilus. Renal vein, which carries blood from the kidney to the heart, and ureter come out of the kidney through hilus. Each ureter is a thin-walled tube. The anterior end of ureter is wide and is called renal pelvis. The ureter opens into urinary bladder. Urine is stored in the bladder till it is filled and then is sent out through a canal called urethra. On top of each kidney, an adrenal gland is present. It is endocrine gland and has nothing to do with excretion. Longitudinal section of kidney. Outer region of kidney is deep red in color and is called as cortex. The inner region is light red in color and is called as medulla. In the medulla are 9 to 12 inverted pyramid-like structures with a broad base towards the cortex and narrow end towards the pelvis. These extend into pelvis. The broad end of each pyramid is branched and gives the appearance of sepals of a flower. Hence, these are called calyces. Through calyces, urine reaches funnel like ducts, and from these into pelvis. From pelvis, urine enters ureters. The cortex extends in between the medullary pyramids as renal columns called columns of Bertini. Each kidney consists of several thousands of nephrons, which are the basic structural and functional units. They are responsible for urine formation. In human kidney, there are about 2 lakh of nephrons. Each nephron has 2 parts. 1. Cup-like structure called Bowman's capsule, and 2. Tubular portion, figure 19.3. Bowman's capsule is a 2-layered cup with space in between the 2 layers. Inside the cup of Bowman's capsule, there is a network of large number of capillaries, which is called glomerulus. Glomerulus, along with Bowman's capsule, is called Malpighian body or renal capsule. After entering the kidney, the renal artery divides into a number of arterioles in the kidney. Each renal arteriole enters a Bowman's capsule as afferent arteriole, forms a capillary network inside the cup-like structure, and leaves the Bowman's capsule as afferent arterially. The tubular part of nephron has three parts. A. Proximal convoluted tubule. B. Distal convoluted tubule. C. Distal convoluted tubule of nephrons opens into collecting tubule. A. Proximal convoluted tubule. The tubule continues further to form a highly network called proximal convoluted tubule. A hairpin-shaped Henle's loop is the next part of the tubule, which has a descending and ascending limb. B. Distal convoluted tubule. The ascending limb continues as another highly coiled tubular region called distal convoluted tubule. C. Collecting duct. The distal collecting tubule of many nephrons open into straight tube called collecting duct, many of which converge and open into renal pelvis through medullary pyramids in the calyx. The Malpighian corpuscle, proximal collecting tubule, and distal collecting tubule of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called as juxtamedullary nephrons. The efferent arterially emerging from glomerulus forms a fine capillary network around the renal tubule called peritubular capillaries. A minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons.